Now it's a pleasure to introduce Professor Ipsita Roy from the University of Sheffield, UK. The Professor Ipsita Roy is a Professor of Biomaterials at the Department of Material Science and Engineering at the University of Sheffield, where she is now head of the Biomaterials Research Group, co-director of the Advanced Biomedical Materials Center for doctoral training, along with the University of Manchester, and she is a steering group member of Roy's Bio, Harry Roy's Institute, and the University of Manchester. She's an expert in microbial biotechnology, natural biomaterials, and their biomedical applications. Her group, currently, works on the production of novel, natural, and sustainable polymers, such as polyhydroxyalkanoates, PHAs, bacterial cellulose, and their biomedical and environmental application. Well, now I leave the floor to Professor Ipsita Roy for her presentation on the production and scale-up of sustainable bio-based polymers of bacterial origin and their use in biomedical applications.
Thank you very much for that very nice introduction. I have to apologize in the beginning because I thought the talk was 20 minutes. So I will leave certain parts away and just take 15 minutes, yeah? Okay. So, like I've been just introduced, I work on natural polymers and mainly of bacterial origin. And if you ask me why bacteria, not that I particularly love bacterial infections, but bacteria can be good. They can produce lovely polymers, and like someone said, in the soil, they can do nice things to plants, they can do nice things to our gut. So I use bacteria in a nice way to produce these natural polymers. So these are my polymers in my lab at the moment, polyhydroxyalkanoates, which have frighteningly worked for about 20 years now. Bacterial cellulose about 10 years, but really scaling up now and really working more on them now. Gamma polyglutamic acid worked in London, not yet in Sheffield. And alginate is the Sheffield baby that we've just started producing in Sheffield. So these are polyhydroxyalkanoates, these are polyesters, and why I've stuck to them for so long is the fact that they have very variable structural properties. So you can have a change in R1, R2, and the X value. So what you're dealing with is really a family of polymers, but not one polymer, one property. So you can really manipulate the properties of PHAs depending on how you grow them, what do you do to them, how you process them. And this is our 30-liter fermenter in Sheffield, so I'm very happy. I moved in 2019, and I have access to this pilot plant we have in Sheffield, and that's where we produce our PHAs now. And when I started years ago, PHAs was already being worked on, so it wasn't a new idea. There was a very big group at MIT, there was a big group in Münster in Germany, so I thought, what should I do? What's new that I can do? I love these polymers because they're green, they're biocompatible, I wanted to work with them. So the first thing I thought, for medical applications, why don't I use gram-positive bacteria for the production of these polymers? Gram-positive bacteria lack lipopolysaccharides, which is a great immunogen, so you don't want an immunogen in a medical polymer, so I thought, let's go with this. The PHA world almost were frightened. They said, you can't produce using gram-positive, they're difficult to break, etc. No, they work. And now there's industrial production using gram-positive bacteria. So this is a typical poly-3-hydroxybutyrate, a polyhydroxyalkanoate, very well known, high melting, quite a high Young's modulus value for a polymer. Elongation at break is not very high, so it's a stiff and nice polymer to work with if you're looking for stiff applications. One of them, in my case, has been bone tissue engineering. And this is a typical fermentation profile. I won't bore you with details. But the nice thing is that now, in Sheffield, we've really been able to scale up. And thanks to ECOFUNCO for the funding for this work, that we really were able to go from a value of about 11 grams to 896, so about a kilo. So I'm really happy and thankful to ECOFUNCO. We change the organism, we change the process, and we're reaching now kilogram quantities of the polymer. Always, I started off with a chemistry degree. We make sure that the polymer is exactly the same every single time. So you saw one of the presentations where they discussed production of PHAs in large scale, and they said variability of property every time it's produced. We get no variability. Every time we try and get, sometimes if the fermentation hasn't gone well, of course. But if it has gone well, we know we've done the right things. It's the same molecular weight, the same monomer content, exactly the same properties. And that's what is important with natural polymers, because if we isolate, we have no control on the properties. So chemical properties, molecular weight, and this is the next type of polymer, the PHAs, the sticky medium chain length PHAs, which the other project was talking about, which I always say we must use them as adhesives. They're very, very sticky and would be a best adhesive to use. Again, this is one of the typical ones I produce routinely in my lab in London. Here you can see it's a low molecular weight polymer. Its Young's modulus is not very high, 0.8 megapascal, but look at the elongation at break. It's really, really stretchable. And it's so really useful for any elastomeric application. So I've used them for soft tissue engineering. Of course, for packaging would be brilliant. This is again a profile. Again, I won't bore you with the details, but I'll highlight, again, thanks to ECOFUNCO, I've been able to increase the yield from about 10 grams, 11 grams, to now 965, almost a kilo. So really, I have a lot to thank to this project, and we hope to get this even bigger as we go forward. Chemical characterization, NMR, molecular weight, we do this every time. And that is the polymer that we've been producing recently, which is a hydroxyoctanoate Co-3-hydroxydecanoate. Again, low molecular weight, elongation at break about 536%, so very elastomeric, low, not low molecular weight, sorry, no melting point, and a very, very elastomeric polymer. What can we do with it? I really am happy that we are able to process these polymers. Very, very few people can really process PHAs. It's not out there yet. Not many people know what to do with it. How do you melt extrude it? How do you make microspheres out of it? We can do it. At a small scale, and I'd like to collaborate and take this to a larger scale, but at a lab scale, we can really process PHAs in many, many different ways, most traditional and the recent methodologies. So I truly believe this is an emerging medical material, and I have to add to this, after ECOFUNCO and all your presentations, I truly believe this is the next eco-friendly green polymer that's out there to be used. We really need to go out there and use it and make the world sustainable. So from the medical point of view, as soon as you say bacteria, surgeons run away from you. But then as soon as you say, no, this is FDA approved already, they come back and speak with you. So this is nice. There is a company out there called TIFA who've already got FDA clearance years ago, 2007, 2009. So this is an acceptable polymer in the medical world. And I've been working on it for a long time, like I said. So many, many different applications. Skin, which I worked with, in fact, beer in polybio skin. Cardiac tissue engineering, very close to my heart, pun intended. Drug delivery, we've done stents, nerve conduits, cancer disease models, really, really lots of work. Bone, cartilage, kidney replacement, and pancreas. And so what have we done? I'll go briefly, I'll look at the time, okay. I've got about 10 more minutes. So bone tissue engineering, one of the main ones that I had started off when I started with tissue engineering with PHAs. This is an example where we gyrated, or pressurized gyration was used in collaboration with Mohan at UCL. And if the video works, wow, it's working. Great. So you can see these nice fibrous structures emerging, like the candy floss you might have seen yesterday at the Luminare. So this is very much looking like that. and look at their structures. This is the SAM. They are wonderfully aligned, really, really nicely porous, so good for lots of different applications. We are obviously looking at medical, but for packaging would be wonderful for certain types of packaging. These are composites with hydroxyapatite because we were looking at bone. And I love animals, so rather than killing animals for the testing of these things, we first look at chicken eggs. This is a particular technology where you can put your polymer or material in a chicken egg, see whether the chick actually grows and is happy and, you know, all functionality is there, and then you take it to an animal model. And I think, I really believe that we shouldn't use animals unless we know that the material is good enough. So this is what we do, and we found, this is all published, we found excellent vascularization and bone formation or collagen formation, and this is then we went into in vivo rat models and got very good results. So this is 3D printing. This is 3D printing of the MCL PHA that we did in London, and I won't go through the whole video. These are the results. So we can really 3D print PHAs, and not many. I haven't heard of anyone doing the medium chain length PHAs yet, so perhaps the first to be able to 3D print them well. And also, obviously, we look at biocompatibility, and green meaning cells are happy, and I'm hoping you agree, it all looks nice and green, and the blue are the nuclei. So this is peripheral nerve tissue engineering. This was my collaborator, John Haycock at the University of Sheffield. And here is when you break a nerve, it doesn't grow and automatically repair very easily unless the gap is really tiny. So what we do is give it a bridge, you know, like the lovely bridges over the river around here. This is a bridge through which the nerves can grow and really repair very easily. These are called nerve conduits. And what happens is surgeons get these nerve conduits, but they're really very difficult to suture. And PHA nerve conduits are beautifully suturable. This is something everybody has told me, and I hope I can get this into the market. So this is the PHA versus PCL. If you see that histogram very quickly, you can see the blue is tiny, and the PHA fibers are really high, showing the high biocompatibility of these materials. And I won't go through all this in details, but we are looking at a dorsal root ganglion. We are seeing whether nerves actually grow into the PHA fibers, and the answer is yes, they do. It's all published, so if you're interested, you can read. We've made these bridges, or these little nerve conduits, and we've gone into animals. Fantastic regeneration. This was in Italy, University of Torino. Like I said earlier, we can't do anything without collaboration, and this is one of those excellent collaborations. We got regeneration of nerve within about 9 weeks. It was exactly the same as an autograft, which is a real nerve. So I think it's got real good potential. Heart, cardiac tissue engineering, and part of a BHF, or British Heart Foundation Center, and the idea is to patch the heart. After a heart attack, what happens is part of the heart becomes non-functional. It's like a scar. When you fall, you get a scar on your knee, for example. But if you have a scar on your heart, not good news. You'll probably have a heart failure very soon. So what we try to do is create a patch, and using particular stem cells called induced pluripotent stem cells, which we convert into cardiomyocytes, and then we hope they will convert into that sort of a cardiomyocyte, which is the mature form. So if we go forward, this was melt electrospinning. I don't know if you're familiar with this technique. It's like electrospinning, but with a melt rather than a solution, and that led to very nice constructs, and this is how they look under the SCM. And what cardiomyocytes do is attach on it, like if we are lying down on a pavement, we'd like to follow the brick or whatever. We don't want to fall off the brick. The cells do the same. They don't want to fall off, so they attach and become long and thin, and that's exactly what we try to do. And this is how they look, absolutely like mature cardiomyocytes. So these results were very good. Lack of time, I'll just skip through them. The other problem with tissue engineering is you don't get vascularization very easily. You know, you need the blood for the nutrition, for the cells to grow. So we added endothelial cells, which lead to blood vessel formation. And these are cardiomyocytes and endothelial cells co-cultured, and then we got really nice cardiac patches onto the heart. So this is the patch onto the heart. This is the heart with the patch on the side. No reaction, no immune response, and very nicely accepted. There is no capsule formation around it either. And that's again the patch with the cells. And the next thing we've done is multi-material, and I probably will not discuss that in detail, just to say it has alginate and PHAs. And the alginate has been used to encapsulate the cells rather than to just add the cells onto the cardiac patch. I'll miss this one out. This is something David had briefly mentioned, that we can really print MCL PHAs beautifully now. They were really flexible. They could make skin, bandages, whatever we need. Medium chain length PHAs, we can 3D print them really, really well. And that's the alginate printing that we can do. And now we've started bioprinting. That is, we put the cells within the alginate and we then print them along with the material. So really speaking, we could go out there and make patches with cells, grow them into any sort of structure we need, and hopefully repair tissue very efficiently. So there we are, multi-material cardiac patch. Many people spoke about antimicrobial in this meeting, so I thought I'd touch upon this briefly. This was a Marie Curie that I led a few years ago now, and we created antimicrobial polymers, including PHAs, of course, with me around, and thio-PHAs. So sulfur-containing PHAs are antimicrobial naturally. They're not produced in a large scale yet. I intend to do so. Hopefully next time when you see me, I would have them in larger scale. But at the moment, we've added active ingredients, so like antimicrobial peptides, and I won't go through the details. Look at the cells. They're very happy on these polymers. I think one thing we have to all remember, if we're doing packaging, fine. If you're looking at anything going into onto the human body, cells, if you're killing bacterial cells with a particular polymer, you can equally kill human cells with the same polymer. So hitting that right balance where you kill bacterial cells but not human cells is very crucial. So we always used to check that, and we got some good results. And these are the thio-PHAs that we produced, and they were very good against Staph aureus, but not very good against the E. coli. And so this is the thio-PHAs that we can print as well. So we always look at processing, because unless you can make something with the polymer, you can't really do much with it. So here are the thio-PHAs that we mixed. This is something we have recently published, which is the selenium strontium hydroxyapatite. This is doped hydroxyapatite. Again, very nicely antimicrobial and really useful as a product, dental application, as a packaging, many, many different applications here. So they work against Staph aureus and E. coli, which is very good news. And finally, bacterial cellulose. So I'll finish with bacterial cellulose. We have produced them nicely. Look at the lovely nanofibrillated structure. That's why I was asking, how do you create the nanofibrillated cellulose? Naturally nanofibrillated from bacteria. Of course, we do all the analysis, make sure it's the right polymer. And we have made antibacterial bacterial cellulose. You know, that sounds quite funny, but that's what we've made it. So you can see here the Staph aureus attaching and dying on this antibacterial bacterial cellulose, cells attaching and growing nicely. These are Hakat cells or skin cells. We've made this nice big bioreactor, and hopefully next time we'll show you some scaled up results of bacterial cellulose. We can print bacterial cellulose now, along with alginate. That's our first printed structure. I'll skip this, but we have done controlled drug delivery, so I was very happy to hear our invited speakers lecture on it. We did make lots of different structures and got controlled delivery. Sorry, I have to talk on the micro. Okay, I move around too much. I hope you could hear me. We've made biodegradable stents. So stents at the moment are metallic with polymers on top, which is not great for the body because the body finds it like a foreign body, not very happy with it. So we are trying to make a biodegradable stent, which was again a project that I led. And we are trying to make 3D models for disease because we always use animals. And, you know, not fair, even if they are mice or rats, they are animals. So why don't we do it in 3D, in a model, check out whether the material or the drug delivery or whatever we are testing works, and then go into animals. So this is something we've done with cancer. So in the interest of time, I think I'll conclude by saying that polyhydroxyalkanoates are really an emerging class of polymers. These are biocompatible, biodegradable. They can be used for medical applications and for ecologically friendly applications. We have used them successfully for hard and soft tissue engineering. Hopefully now we'll start using them for eco-friendly applications. Bacterial cellulose is yet another natural polymer with a huge potential, both for biomedical and for packaging, as we heard today. So I'd like to thank the numerous lab members, my collaborators, and the funding, of course, ECOFUNCO, a prominent one for this talk, and of course, my lovely group. And thank you all for your attention.
Thank you very much, Ipsita, for this interesting overview of the biomedical applications of PHAs. So, any questions? Yes.
Thank you very much for a lovely presentation. I know you had to go quickly through it because of the time, but can you elaborate on the 3D printing of bacterial cellulose? Because I thought it was quite difficult due to the structure of the fibers.
It is. So we've literally done this last month, so not much of detail there, but what we did was used alginate almost as a binder. So it's very nice if you take the pellicle, mesh it, really make a mash of it, and add it to alginate and then co-print it. And they see it's not the best, but you saw the structure was quite stable. And of course you need the cross-linker there, but we're using alginate to keep it nice and stable.
We need to talk.
Absolutely. Come to Sheffield.
Thank you, Ipsita, for your nice presentation, and congratulations for such nice results. About the 3D printing of bacterial cellulose, because you also mentioned that you used melt electrowriting for PHAs. Do you think if you can use also melt electrowriting of BC, bacterial cellulose?
I don't see why not. Yeah, absolutely. Unfortunately, I don't have a melt electrospinning equipment yet in Sheffield. I used to have one in London, and we're buying one, hopefully in the next month. I don't see why not, because it has high melting. We've checked the TGA values. It doesn't degrade until quite a high value, so we should be able to do it. It depends whether we can do it on its own, or we need some sort of binder like we do in our 3D printing, because we're using alginate. So perhaps, again, an alginate can be used as a binder, but I don't see why not. Yeah, sure.
And my second question, what was the differences between the structure of PHA you produced with 3D printer and melt electrowriting? Because you had also different layer with melt electrowriting, as I saw. What was the main
difference? Very good question. Of course, as you know, the technique, you can get much finer structures with melt electrospinning. So the struts that we got were about, I think, 100 microns. When we use 3D printing, although we use very funnels or nozzles with very small pores, but getting to about 300 is okay. Lower than that is a bit tough. Whereas here you can get much thinner struts.
Okay,
I think that's the main difference.
Thank you.
Thank you. Yes, can I go?
Yes.
Oh.
Okay. I have some curiosity, Ipsita, about the production and recovery of PHAs from your bacterial biomass used. So I'd like to know the average content of PHA accumulated in the bacterial cells and the extraction and recovery methods used to extract.
You've hit it absolutely at the right point. So I don't like using genetically modified bacteria. So companies like Metabolics, or most of the companies use genetically modified strains, and those can go up to 98%, dry cell weight of the polymer. I like to use wild type for safety purposes, and also you have to add an antibiotic to the fermentation, adds to the cost and all that. So the highest we've achieved, which I'm very happy with, is 80 with a wild type, Putida. So I'm very happy, and again, I keep thanking ECOFUNCO. I couldn't have done it without the funding, but we've achieved 80% dry cell weight with HOHD. At the moment with HB, we are only 38, but for a wild type, it's quite high, getting close to 40. Extraction, I'm not happy with my process at the moment. I'm doing sock-slit extraction, which is great because it's medical grade. I never get any reactivity in animals or anything, but it's not a scalable technology. So I really want to work, like I keep speaking with Cellulosic here, and others. So I really need to change the extraction process and make it scalable. That's my next step forward.
Thank you very much for your
Thank
presentation again.
Maurizio, sorry, excuse me. We have another question from one of our Zoom attendants. And if you, Ipsita, can explain something more about PHA for drug delivery.
Of course. Sorry, I couldn't really talk about it here, but we've done quite a lot of work with drug delivery. So we've made microspheres, and the average diameter we've achieved is about, I would say, nearing a micron. We can get there quite easily, or half a micron. We've started now looking at making nanoparticles as well. We get really lovely controlled delivery, and we mainly use poly-3-hydroxybutyrate. We haven't used the MCL PHAs in a big way. One of my postdocs had done some work, and they also can be used, especially as a blend. You can get double-layered microspheres with HOHD and HB. These are emulsion, so we make these emulsions and we make the microspheres, and they are very, very controlled in terms of delivery. You can target them. For example, I've targeted them to cancer tissue. So on the outside, I've put the receptors that would go and target just the cancer tissue. So when you do chemotherapy, for example, for patients, they die because of the chemotherapy, really, because you're killing all the good cells and the bad cells, which doesn't make sense. So if you can have targeted delivery of the anti-cancer compound, that would really make their lives much easier. And that's something we have done as well.
Thank you.
Okay, thank you very much.